fluke and tapeworms, control and prevention. Problem of parasitism, endoparasitism is concentrated most of the time with the gastrointestinal nematodes. So it is time that we also focus on flukes and tapeworms because they are also as responsible for the economic loss and other drawbacks as much as the gastrointestinal nematodes like Haemococcus. Now, before going into the details about these flukes and tapeworms, let us first know what is the basic difference between flukes and tapeworms. Now, flukes, though both of them belong to the family Platyhelminthes, they are both flatworms, but they are quite different in their morphology as well as their egg structure. Flukes are generally mostly leaf-shaped. Their length is quite lesser. They are smaller. Even the larger flukes are about 4 to 5 centimeter only, but tapeworms are longer, larger. They are ribbon-like. That's why known as tapeworms, and they can extend up to 4 to 5 meters even. And one of the major way of distinguishing both the types of parasite is that the tapeworms are segmented. They have a head, they have a neck, and they have segments, mature, immature, as well as gravid segments. Apart from that, 90% of the tapeworms, 80 to 90% of the tapeworms mostly inhabit the small intestine, while in the case of flukes, they are found in many other organs like liver, lungs, rumen. As far as the eggs of these parasites are concerned, fluke's egg are oval in shape, and a peculiar thing is that at one end, at the anterior end, they have a cap-like structure known as operculum. But in case of tapeworm eggs, we'll find two types of tapeworm eggs. Either the Anocephalidae family eggs, that means your Monigia, Stilicia, and all that are mostly affecting the small ruminants. They are usually either triangular or rectangular shape, and they have a peculiar pyriform apparatus inside it. And in case of Tenidae family eggs, they are rounded in shape and they have a hexacanth embryo. There are 3 pairs of hooks are there inside it. So this way we can differentiate the two. Now coming to flukes, and one of the very important flukes are the fasciola species. In India, Fasciola gigantica is more prevalent than the other Fasciola hepatica. And though Fasciola hepatica is there, it is mostly concentrated to hilly areas. And this is the prevalence is based on the availability of the intermediate host. Though the intermediate host is snail for every species, the species of snail varies. And they are the liver flukes. They are mostly found in the liver and bile duct. Actually, for knowing the control and prevention, it's very essential to know the life cycle of the parasite. In order to control the parasite, we need to interrupt the life cycle of the parasite. So the life cycle of this fasciola starts when the adult parasite, which is found in the bile duct and liver, they shed eggs into the bile, which enters the intestine, and then the eggs pass through the feces. So after the eggs pass through the feces, these eggs hatch. The hatching of the eggs to release a motile form known as miracidia that takes 10 to 14 days. After this hatching of the egg, then the snail, which is the intermediate host, this comes into play. And the thing is, miracidia needs within 3 hours, the miracidia needs to penetrate the snail. Otherwise, the life cycle will remain incomplete. So after penetrating the snail intermediate host, this miracidia will now change into different other forms, sporocyst, then Anidia, and then Cercaria. From the snail, now the Cercaria will move out. This Cercaria, which is also having a tail, they insist on the plants and herbaceous. And in this plant and herbaceous, they turn to the infective stage. Now, this is the stage which we need, the metacercaria stage that will infect the host, which is a sheep and goat population. On ingestion of this metacercaria, which is there in the plant or herbaceous or grasses, this ingestion of this infective metacercaria which is the infective stage. This will lead to the start of the infection. Another interesting thing is that infection of snail with one cercaria can produce 600 metacercaria. So once the metacercaria is ingested by the animal, this metacercaria, they exist in the small, after ingestion, they will exist in the small intestine. And from the small intestine, they migrate through the gut, crossing the peritoneum, and then enter the liver. Now, another thing is, though they enter the liver, they migrate in the liver for 5 to 9 weeks. Now, during this migration period, they produce migratory tracts, and this time, this fluke is not totally mature. It is still a juvenile fluke, immature fluke. So after 6 to 8 weeks, it then goes to the bile duct, and the bile duct is the place where it gets matured to adult form, and it occurs only after 9 to 12 weeks of infection. After 9 to 12 weeks after the animal has ingested the metacercaria, the adult form is present in the bile duct. And when the adult form is produced, we can see there are two important clinical manifestation of this disease. One is the acute form and another one is the chronic form. The acute form is the form when large number of metacercaria ingested in a short time, and in this form you'll find number of immature or juvenile flukes. And here acute form, since large number of metacercaria are involved, there will be rupture of liver capsule, and the sheep actually die within a short span of time. They die. The symptoms you can see is blood stained growth in the nostril or anus. Then there will be also since this fluke causes blood loss, they suck blood from the animal about 0.2 ml. So they anemia is there. Each fluke sucks blood, and if large number of flukes are there, anemia become apparent. And during post-mortem in this condition, you'll find that liver becomes enlarged, as there will be inflammation of the liver, and liver becomes enlarged. Then there will be also, due to this migration of immature fluke in the tract, there will be tracts of this migrating fluke, then blood stained fluid in the abdominal cavity also. Now a more common form is the chronic fasciolosis. Acute fasciolosis is a bit rare than the chronic form. In chronic form, there will be progressive anemia will be the loss of condition will be there. Then a important clinical sign is the bottle jaw condition, which will be seen due to hyperproteinemia. Then we can see that the flesh of the sheep becomes brittle, debilitating condition is seen. And on post-mortem, you'll find that the liver is there is damage to the liver. And also we can during post-mortem, if you got the bile duct and the liver also, or section of the liver, there will be adult flukes you can find. So in the post-mortem, actually, you could have seen that the whole of the carcass, when it is cut open, abdominal cavity is full of blood, because there is hemorrhagic hepatitis and hemorrhage into the peritoneal cavity. And due to that, the animal dies within very few days of onset of the disease. Now, another very important infection and very common also is the amphistome species. Amphistome is a group of flukes that known as amphistomes, and it contains a lot 7 to 8 genera of amphistome species like Paraamphistome, Stomum, Fisidarius, Cotylophorus, and like that. You can see it looks like a pomegranate seed, and it is attached to the rumen, commonly known as rumen fluke. They have quite a lot similar to with the life cycle, just like the liver fluke. Here also, the amphistome have the similar type of life cycle. The eggs, they are released from the animal body through the feces. They turn to miracidium within 1 to 2 weeks. The eggs hatch and turn to miracidium within 1 to 2 weeks. And then they enter the snail, inside the snail within 4 to 6 weeks. And here the snail are the Indoplanarbis. The species is Indoplanarbis. It is a water snail, and in that snail also other some more stages are developed, and the final stage that comes out of the snail is the cercaria. You can see they have a tail-like thing which helps in the water. They come out through it with the help of this tail, and then again they turn to metacercaria in the grasses, and where they encyst. This metacercaria is actually viable for 3 months. This metacercaria exists in the duodenum, feed for about 6 to 8 weeks in the duodenum. Now, in the duodenum, now this is also immature form of the amphistome, which is in the duodenum. And during this period, maximum damage to the animal occurs because these immature flukes are the actual pathogenic ones, because they are plug feeders. That means they eat the plugs of mucosa. The young flukes, they draw pieces of mucosa, and this mucosa of the intestine, they slough up this tissue, causing necrosis and hemorrhage. And due to this immature amphistomosis, it's pathogenic, and it's clinical and has a clinical manifestation, which we'll talk later. After this immature amphistome is in the duodenum for 6 to 8 weeks. After that, they move to the rumen, and in the rumen they turn to adult 2 to 4 weeks of coming from the duodenum. You can see it is these are the immature ones inside the intestine, and these are the mature flukes in the rumen. Now, the adult amphistomes are actually well tolerated even when hundreds or thousands of flukes are present. There is hardly any big loss or big damage to the rumen. Only there is some detonation of the ruminal papillae, but the main damage is caused by these immature amphistomes. As it feeds on the mucosa of the intestine, and as it draws the mucosa plugs and cause hemorrhages, there is anemia, there is hypoproteinemia and emaciation in the animal. When we do post-mortem, the contents of the intestine contains blood and also some mucosal sticks. Actually, in case of mature fluke, there is hardly apparently no clinical signs is there. Whereas in case of this immature fluke, there will be anorexia, and due to high fluid intake, polydipsia is also seen, and due to decomposition of the proteins in the intestine, there is very foul smelling foetid diarrhea. This is very important. Also, due to this continuous diarrhea and due to rectal hemorrhage, due to prolonged staining may be there. There may be also soiling of the hindquarters and also tails with this fluidy feces. This diarrhea is also known as shooting diarrhea because of this projectile motion of the diarrhea. It is essential to treat animal which void amphistome egg, even when they do not show clinical signs. See, many a times this mature form, they don't show. If mature form is there, no clinical sign, and clinical signs are not very apparent. But still, it is essential to treat the animals because these eggs are a source of infection for the snail intermediate host, which will again infect new animal, new host. So it is very essential to treat animal which void amphistome eggs. Liver fluke and rumen fluke, both amphistome and fasciola, there is every chance of co-infection. And the problem is there is many times there is misdiagnosis of this infection because of the similarity in the type of eggs. But the problem is the treatment of these two flukes are quite different from each other. Not all anthelmetics work for rumen flukes. There is very limited anthelmetics for rumen flukes. So it is very essential to properly diagnose the infection, whether it is rumen fluke or it is liver fluke, and then accordingly treat. So to differentiate the eggs of fasciola, which is find diagnosis of these helminths are the fecal sample examination is the gold standard, and most of the field veterinarians should go for fecal simple examination before treating the animal. And there is always confusion with fasciola and amphistome eggs. So to differentiate the fasciola and amphistome egg, it is essential to know that the fasciola egg, it has a yellowish or golden brownish tinge to it. Amphistome egg is quite clear and transparent. Another thing is that in case of amphistome egg, the inner embryonic masses or division, they are very distinct. Inner divisions in case of amphistome is quite distinct than the fasciolid eggs. And also there is a posterior knob in amphistome, but it is not because we see a lot of sample, you may not see the posterior knob in many samples. So it is better to differentiate basing on the embryonic mass as well as the color of the egg. And those who are experienced, they will know that amphistome egg is bit larger than the fasciola egg, though not much difference is there. It is a bit larger than the fasciolid eggs. There is another fluke, dicrocilium, though it is not present, though its presence is not pan-India. It is mostly present in higher altitude hilly regions, is the Dicrocilium dendriticum. The egg is quite smaller and it is flat in one side. Here there is involvement of two intermediate hosts. One is the land snail and another is the ant. And it is relatively less pathogenic than fasciola, though it affects the liver. It is lancet liver fluke, though it affects the liver. It is relatively less pathogenic. In case of heavy infection, there may be some cirrhosis in the liver surface and some production. There may be some reduction in the production traits of the animal. And here we can give albendazole for treatment of this fluke. The burden of trematodes, mostly the amphistome and fasciolids, is dependent on many factors. It is influenced by the climate. It is influenced by the availability of pasture. So the drug that has to be given to the animal should be active against both mature and immature stages, because immature stage of the parasites are also as harmful as the mature stages. Now for liver fluke, triclavendazole is proven to have an effect both on the mature and immature stages of the parasite. Closantel is said to be effective against the earlier stages of the fluke, but mature stages it is having lesser effect. And also oxyclosanide is capable of only killing the adult liver fluke, not the immature form of the fluke. While in case of rumen fluke, oxyclosanide is highly effective both against the mature and immature fluke. Niclosamide is having more effect on the immature fluke and the adult fluke. So this is about treatment, but if we come to control, as we can know from the life cycle of the fluke, since snail is the intermediate host, controlling the snail population is also a very effective way to control the infection or the chain of the infection. So intermediate host control is important. And since the snail multiplies so rapidly, it is not possible to totally eliminate the snail population, but still there are some ways in which the population can be reduced to, or the infection or contact with the snail population can be avoided. The male snail habitat is mostly water bodies, lakes, and mostly the peak of snail season is at the end of the rainy season. And as soon as the water level is reduced, male population is also reduced, but infection level rise because animals congregated in this area. For control of snail population, we can fence the area, that is snail infested areas can be fenced. Then low-lying area should be proper drainage should be there. And since snail multiply in wet areas, draining the marshy pastures should be able to reduce this population. And also plowing of the soil to a depth of more than 5 centimeters expose the snail egg to sunlight. And due to this, it is advocated to plow the soil to reduce the snail population before sowing. Another very important aspect is grazing management. So to provide clean pasture to the animal is very, very important. And for that matter, zero grazing, as we call it, it is a very effective way for controlling the infection due to flukes. And there can be other ways like processing of fodder. If fodder is collecting, is suppose the fodder is collected from water stagnant areas, then the portion above the water should be used, should be used for filling. And if we are using the portion under water also, that should be dried, that can be used as hay, because Metacercaria cannot withstand temperature above 28 degrees. So if it is dried and used as hay, then the chances of infection reduces or lessens. Now, there are also some chemicals that are used for snail control, which is known as molluscide, like copper sulfate, copper pentaphenate. But in most of the countries, due to environmental problems, environmental effects, they are not been advocated as such. Now, animals, when an animal is coming to farm, and all types of animals, because they share the same pasture, they should also be included in control program to avoid, because we need to avoid the animals that are already having heavy burdens of this parasite, or they are, and there is now the problem of drug resistant flukes have also come up. So to avoid that, it is advisable to include all the animals coming into the farm, if there are multi-species are there, into this control program. Then effective use of drugs is also very important. Accurate dosing is also very, very important. And a single product should not be used, because it will lead to development of resistance. Then it is essential not to overstock animal. The grazing pasture should be such that there should not be crowding in the pasture. Nowadays, there has been research on biological control. Verticillium chlamydosporum and Ponchonia chlamydospora, they are grown in the laboratory, and they're the nematophagous fungi. They have also effect on the helminth eggs like fasciola. A lot of research is going on using plant on herbal extract. Some rural areas also, a lot of plants are being used, like allium, piper, erica papaya. Now coming to the tapeworms, there are few tapeworms that affect the small ruminants, as we can see, Monagia, Avitilena, Stylicia. Apart from Monagia and Stylicia, the other species are relatively less pathogenic. They don't show much clinical signs, but if they are present in large numbers, and since they are present in the small intestine, they may occlude the small intestine if present in large numbers. Most prevalent tapeworm is the Monagia species. And the Monagia species, the life cycle of Monagia species is quite simple. Simple than the flukes that we recently talked about, because the larval there are few larval stages. There is only one larval stage, and after the animal defecates and the eggs come out, and the gravid segments containing the eggs come out, they are ingested by this intermediate host, which is the oribatid mite or the forage mites or the pasture mites. And these mites, they are present in the pasture, and when the animal ingests these mites, they get the infection. Gravid segment, like gravid segment at the segments present in the posterior end of the tapeworm, and when they are passed, monagia, especially monagia segments, they look like cooked rice. Also, many times you can see the animal passing an entire tapeworm, a long string. And you can, I've already discussed the tapeworm eggs. They look like this. They are triangular, mostly triangular in shape. Sometimes they are also rectangular, and there is a pyriform apparatus inside it. So these monagia species are also, it is mostly pathogenic in young animals. Present in lesser number, they may not show any, there may not be any clinical manifestation. But if present in more numbers, they cause unthriftiness and lot of GI disturbances. The easy way to diagnose this is, most of the times farmer can see the tapeworm segment in the dung itself. Impact of the tapeworms may lead to some secondary disease like enterotoxemia. Even in case of acute fasciolosis, there is chance of growth of this, there is Clostridium novi black disease due to anaerobic condition created by these parasites. Of the anus of the, you can see really 2-3 meters of tapeworm. They're coming out from the anus of the ruminant. So light loads of tapeworm doesn't seem to be a problem, but severe infestation can cause problem. And the time taken for the adult to, after ingestion of this infective form, the time taken is about 6 to 8 weeks. And this tapeworm in high numbers, as I told, can block the intestine and also affect the gut motility. In case of Stilecia punctata, which is the tapeworm seen in intestine, there is another Stilecia species which is seen in liver and bile duct. This Stilecia punctata can cause some nodules in the jejunum region of the intestine. Coming to the treatment of this tapeworm, praziquantel is effective against both the adult and the immature stage of the tapeworms. Also, benzimidazole group of drugs can be given, among which albendazole is the most effective, is considered the most effective, and but it should be given at high doses only. That is more than 15 mg per kg body weight. Fenbendazole can be given, but if only if the dose is doubled. Here the praziquantel is the drug of choice. And goats require higher doses than sheep. It is because of the higher metabolism in case of goats, and also because of the drug bioavailability. There are also some old remedies that can be used against tapeworm infection, and this includes the use of pumpkin seeds, pomegranate bark, and powdered areca nuts. There is also need to plow and reseed the pasture. We should always try to avoid the use of same pasture, especially for young animals, because in young animals, this monagia infection is very much common and pathogenic mostly in young animals only. The infection with bladder worms, though definitive host is not sheep and goat, in case of bladder worm infection, the primary host is not sheep. Unlike monagia, unlike fasciola, infection with certain tenia species of parasites in sheep and in dog, especially canine tapeworms, they have sheep and goat as intermediate host. And interestingly, the adult parasite cause not much pathogenesis. They are relatively harmless in definitive host. It is the intermediate host, that is the sheep and goat, that is actually harmed. So bladder worm cyst, they are actually the stages, the infective stages, and sheep and goat acquire the infection when they ingest the tapeworm eggs, the canine tapeworm eggs on pasture. And once this egg, they ingest the egg and the egg hatch in the body, the larva that comes out of the egg, the oncosphere, they migrate through the gut wall and travel through the liver or other organs, and they form a cyst-like structure. This is the life cycle. So dog is the definitive host. Sheep and goat are the intermediate host. From the adult parasites are present in the intestine of the dog. The egg the egg are being taken up by the ruminants, small ruminants. Inside the small ruminants, the different organs, they form this cyst ladder worms. And when the definitive host again comes in contact with this cyst, ingests the cyst, they will get the infection, and this cycle will go on. So very important, cysticercosis or Cysticercus teniculus. The main parasite, the main tapeworm is Taenia hydatigena. So it is a cosmopolitan parasite of dog found in the intestine. And this Cysticercus teniculus is actually a loosely filled cavity having a neck, and they are mostly found in the abdominal region, attached to the abdominal cavity, and also to the livers of the animals, mostly sheep. So the effect of this Cysticercus teniculus is that having these cysts in the liver and other organs, they lead to condemnation of the meat, and which impacts the economics, and it leads to the economic loss. The metacystole also serve as a predisposing cause to black disease and may lead to also traumatic hepatitis. So outwardly, the sheep shows no clinical signs. There is no clinical signs as such. Many a times, only on slaughter, we can find the cysts. The cyst, you can see it is a balloon-like watery fluidy cyst having different diameters. They may be present in single or in more than single, and mostly they affect the abdominal organs as well as sometimes also liver. They are also known as false hydatids because they have some resemblance with the hydatid cyst. Now, this infection with this tenuculus larva can look like acute acute fasciolosis, because they have the blood filled, there is blood hemorrhage, that is abdomen is filled with blood. So, but there is scar formation also, and there is also liver, but this presence of this cyst will give away the condition. So the sheep or goat that act as intermediate host, and the cyst inside, after ingestion of this egg, the cyst is formed almost 7 to 10 weeks after the ingestion, and after which they degenerate. Now this degeneration may precipitate sudden death, and that may be the clinical presentation at the end. Now this treatment of this condition is praziquantel, but the dose is comparatively higher than Monagia and other tapeworms. It is at 50 mg per kg body weight. Another important is the Cinerus cerebralis, which is and the primary tapeworm is the Taenia multiceps. It infects the brain and the spinal cord of the intermediate host, while in the primary host it may not cause any damage. And there is neurological signs can be seen, since it is affecting the brain and spinal cord of sheep, the neurological signs, and that also depends on the location of the Cinerus cerebralis. If it is in the surface of it is in the brain surface, then animal holds its head high and tilts to one side. If it is in the anterior part, then the animal will step high. There is head pressing is there. There is irregular gait will be there. There will be head in coordination in moving, head inclination will be there. Then the feasible treatment is the surgical removal, and only if the cyst is accessible. Mostly if the cyst is present in the surface of the surface, then maybe if it is accessible, then only some type of surgical treatment can be done. Otherwise, there is no such treatment as such. These are the various forms of this, which is also commonly known as GID or staggers. So this ruminants, again, the mode of infection of this is again the same way, is by grazing, by eating the egg containing the oncosphere. And here also the same, dog is the major host, but small ruminants develop immunity to reinfection. And also this immunity depends on the immune status of the animal. And after immunity, even after having immunity, the cyst can remain in the tissue for a majority of its life. There is another bladder worm which is known as sheep measles. The common name is sheep measles. Here the size of the cyst is comparatively smaller. They are oval. This is also fluid filled, but the cyst remains viable only for a short period of time, almost 6 weeks only, after which the larva dies and the cyst becomes calcified. In case comes up in the abattoir, then either the cyst must be trimmed from the muscles, because they insist in muscles, so they may be trimmed from the muscles. But if we find more than 5 cysts, then the carcass has to be condemned. The muscle has to be condemned. Hydatid cyst is a infection which is having a lot of public health and zoonotic importance. Here also dog and wild canids are the definitive host. And this adult tapeworm in the dogs are rather harmless. You can see the harm is actually caused in the intermediate host, that is sheep and goat. This hydatid cyst also, due to accidental ingestion of infective egg, leads to infection. And the organs that are targeted, that are most in which hydatid cysts are mostly seen, are the liver and lungs. So both in sheep and goat, the primary sites are the liver and lungs, but it may spread to it may go to other organs like spleen, heart, brain also. Here the eggs pass. Here why this hydatid cyst is important, because the eggs passed by dog actually can remain viable on a pasture of 2 years. And also this hydatid cyst grows slowly and takes several years to develop. There may lead to displacement if the size becomes more. If it grows, the size of hydatid cyst becomes more, they may cause displacement of lungs or liver tissue and fibrosis of the area surrounding the cyst as well, and also put some pressure on the organs, leading to different pathological tissue changes. The effect of the cyst on this host, on small ruminant, depends on the size and location of the cyst. In very rare case, there may be rupture of the cyst into the serosal cavity, which may lead to fatal anaphylactic reaction also. The control measure for this hydatid cyst includes, and also other cysticercosis, includes how is interrupting, breaking the cycle between the definitive and the intermediate host. That is important, to break the cycle between the definitive and the intermediate host. That's why the control measure includes to prevent the dog from accessing the infected row of fowls. There should be proper disposal of this hydatid cyst. Also, the sheep, it is important to prevent the sheep from getting infected by the eggs of these tapeworms. That can be done by proper disposal of the dog feces. controlling the movements of food animals and dog, and other controls. Also, there should be control of population, stray dog control. Raw sheep or goat meat should not be offered to the dogs. Meat should be frozen, should be kept at minus 10 degrees centigrade or lesser. There should be burning or burial of the condemned meats, and if it is going to be used as dog food, there should be proper sterilization of the offals. And also some hygienic measures should be taken so that the infection does not spread from the dog to the small ruminants. So the thing is, even after a control program is there, since the pasture is having viable eggs, it is very important that pasture management is very important. That pasture should be, there should be clean pasture, and most probably not probably, there should be focus on rotation of the pasture, and also zero grazing should be there should be effort for zero grazing. Though this liver scarring are present in sheep for is life, and also the life cycle, though it has been broken, there is every chance that again the life cycle will continue because there is presence of eggs in the pasture. So control of also dog population, and since wild carnivores are also primary host, the control of this population can also aid in prevention of this infection. So this is a summary of some of the important flukes and tapeworms, their life cycle, their pre-patent period, their important aspects of these parasites. And we can see that in Monagia species, the young animals are affected. The diagnosis of these parasites is easier because we can find eggs also. Fluke eggs can be diagnosed by seeing by microscopic examination, and also the control of these flukes and tapeworms relies on breaking the life cycle of the parasite by proper grazing management, by making sure that our animals do not come in contact with the intermediate host, like snail in case of flukes or other, and which they gain access during grazing only. So grazing management holds a very important aspect of control of these parasites. Not just treatment and control, prevention of reinfection is also very vital. Because as I told, in case of phytitis cyst, since there is presence of viable eggs or viable infective stages in the pasture, it is very important to prevent reinfection. The proper management aspect is important. Diagnosis is important for treatment and control. Also prevention. For prevention, we have to go for proper grazing management. When it is possible to do postmortem, it is very important to do postmortem, because on postmortem aids in, it is very important to, it also aids in diagnosis. Many times the carcass has to be opened except in case of some infectious disease like anthrax, because some acute infection, in some cases there may not be clinical manifestation. In acute infection, there is chance of not getting the actual causative agent. So postmortem examination should also be a vital part of diagnosis. Thank you.